Hi, my name is Niklas Hohmann and I will be talking about how to estimate disintegration risk of skeletal remains. Deposition rate and the decrease of disintegration rate are major drivers of preservation of shells, and since the preserved proportion of shells determines how much we can infer about the ecology from the fossil record, I will be looking at the disintegration rates today. The aim is to develop a method that allows to estimate skeletal disintegration rates, and I would like to do that locally to capture the spatial patchiness of taphonomy. I would also like to do this taxon specific, just to get an idea of how much disintegration rates vary between different taxa. And I would also like to know how the disintegration rates change with the depth in the sediment. So I would like to know what is the shape of the taphonomically active zone, and I would actually like to assign a disintegration rate to a specific depth in the sediment. For this, I use some data from the Northern Adriatic Sea from the 4 publications listed here. These are all amino acid dated shells of 3 different taxa, and overall these are more than 1,000 individual shells with assigned dates, so this is a really cool data set. As an example, here you can see one of the age depth distribution of shells, giving the age of each shell and at the depth at which it was found. But here one thing becomes very clear. Even at the same age cohort, shells are extremely mixed, so we find shells very close to the surface and almost a meter or more buried below in the sediment. And this has some very strong implications, just because the speed of burial is thought to be an important driver of preservation. But at the same time, we find shells with extremely varying speeds of burial, even in the same age cohort. And to incorporate this changing speed of burial between individual shells, I have developed a taphonomic model. This taphonomic model is called the AALPS model, which stands for Aging Along Burial Paths. And it is based on the concept of taphonomic age, which is, simply said, the damage a shell has accumulated over its lifespan. I'm going to use one analog for this, which is radioactivity. Because what matters about radioactivity is not like how much a person is exposed at a specific time, but how much exposure of radioactivity does it accumulate over its lifespan. And this will then determine the likelihood of getting cancer, for example. We need 2 types of information. The first is the disintegration rate at a given depth in the sediment, and this will simply determine the shape of the taphonomically active zone. The second part is the shell depth at a given time, which describes the burial path each individual shell has throughout its lifespan. Combining these 2 will give the disintegration rate at a specific time that one individual shell experiences. By accumulating all these values, we get the taphonomic age of the shell, which is simply the exposure to disintegration rate the shell has experienced up to a time t. This idea is actually quite old. As an example, Flesser mentions a so-called taphonomic clock, which is a clock that runs faster close to the sediment surface and slower deeper down in the sediment. And then taphonomic age is essentially the age of a shell measured on this taphonomic clock. Here as an example, a age depth plot with 2 burial paths, and the disintegration rate, a very bright yellow indicates a very high disintegration rate, and a red indicates a lower disintegration rate. Both of the shells end up at almost the same depth, but one spends a lot more time very close to the surface, whereas the other one is buried quite quickly. And looking at the taphonomic ages of these models, we can see that the one that spends a lot more time close to the surface ages a lot faster. And the taphonomic age also directly translates into preservation probability. Taphonomic age has some really cool properties. For example, it directly translates into preservation probability. And the lifespan of a shell in taphonomic age will always follow an exponential distribution. So the average lifespan of a shell measured in taphonomic age is always 1. The model is strongly dependent on the burial path of an individual shell, but we have so far not made any restrictions to this. To make the model usable, I will make some model assumptions about how these burial paths look like. For this, I will assume that if shells are found slightly shallower than predicted by the age model, they have simply been buried slightly lower than the average deposition rate, whereas if they found deeper than predicted by the age model, they have been buried slightly faster than predicted by the average sedimentation rate. This allows to reconstruct a burial path based on the age model. But there are some really important questions still open, and one is that what assumptions on the type of burial paths are actually plausible? And if we have a model about burial paths, how can we test it against empirical data? If anyone has an idea, I would be really happy to get feedback on these questions. With all this in place, we can now start looking at the model and its behavior. For example, here I assumed an exponential decrease in disintegration rate downcore, and this is the type of taphonomic age with age and depth I get. And as expected, the taphonomic age in the upper right part is pretty high because these are the shells that are close to the surface for a long time and therefore exposed to a lot of destructive processes, whereas at the young and deeply buried shells have a very young taphonomic age because they essentially have not been exposed to any destructive processes in their lifespan. Translating this into preservation probability, we see the same thing. Shells that are buried extremely quickly have a very high preservation probability, whereas shells that spend a lot of time close to the surface are very unlikely to be preserved. Now I come to the main focus of the talk, which is estimating disintegration rates. And this is only possible under one additional requirement, and that is that the shell assemblage is actually well mixed. So all taphonomic ages are approximately equally likely. And the estimation is based on model fitting, and that is just matching taphonomic ages predicted by the model against the exponential distribution expected from the taphonomic ages. And in this case, I use it to estimate parameters of different parametric models. For this, I selected 3 hypotheses about how shell disintegration can change downcore. The first is that disintegration changes exponentially downcore. The second hypothesis is that it changes stepwise. This has the underlying assumption that there in the surface mix layer there's a very high disintegration, which then drastically drops once there's less and less bioturbation. And then there's also a combined model, which is just a combination of the assumption that bioturbation has a strong influence on preservation, and then once bioturbation decreases, we get a slow decline. As raw data, I use the core from Panzano, and this is the age depth distribution of the shells from this core. And these are the results of the model fitting procedure based on the AALPS model. Here, the p-values are for the null hypothesis that this is a good fit, so a high p-value is actually desirable. As the exponential model has the highest p-value, I will now only focus on this model. And these are the taphonomic ages of the corresponding skeletal disintegration rate. And this is the preservation probability derived from the exponential model. Again, it is clear that shells that remain close to the surface for a long time have a very low probability of preservation, whereas all shells that are rapidly buried are most likely to be preserved. of course, there are still a lot more open questions. One thing I would like to look into is trying to incorporate the uncertainties that are involved with the dating, for example, the amino acid dating or the lead-210 dating that is underlying the age models. The next thing is trying to figure out how to reconstruct primary shell input based on the skeletal disintegration rates we estimated, which is of course of great importance for ecological studies. And as an outlook, I would also like to look into a sequence stratigraphy perspective, trying to figure out how to get an idea of time-dependent disintegration rates if taphonomic conditions change within the different system strats. To sum this up, I presented a model for skeletal preservation, and it is based on individual burial paths and the idea of a taphonomic age, and this allows to estimate skeletal disintegration rates based on the age depth distribution of shells. But there are some really interesting open questions, and one of the most pressing is how regular are these burial paths actually? Because this has strong implications for the preservation probabilities the model returns. And how do the conditions of sedimentation and bioturbation actually translate into these burial paths? So if anyone has any feedback on these questions, I would be very happy to discuss them. Also, I would like to thank Emilia Jarochowska, Rafał Nawrot, Teresa Nohl and Michal Kowalewski for their feedback on this research. And if you have any questions, just write me an email. Thank you.